Welcome to another lecture by MedicoMedics, Anatomy Chapter 12, the Immune System. In this lecture, we will provide an overview of the functions of the immune system, its components. We will compare innate versus adaptive immunity, talk about antibodies, the lymphatic system, the immune response, clinical relevance, such as immune disorders, briefly mention vaccination and immunization, diagnostic tools, and end with a summary. Now, before we focus on the anatomy of the immune system, meaning key structures, let's talk about the functions of the immune system. So its function is to protect the body from pathogens like bacteria, viruses, fungi, parasites. Another function is to identify and eliminate abnormal cells like cancerous cells, remove dead or damaged cells. Another function is in generating immunological memory for faster responses to future infections. Now let's get an overview of the main components of the immune system. So primary organs include the bone marrow. This is where immune cells are produced. Now you find bone marrow, as illustrated here, inside the medullary cavities of bones. And if you want to be more specific, in axial skeleton, you can find it in vertebrae, sternum, ribs, skull, and pelvis, especially in the iliac crest. You can also find it in the proximal ends of long bones, for example, in proximal femur and humerus. In children, though, you will find it mainly in the diaphysis of long bones. So you will have red marrow present in the shafts of long bones, but is gradually replaced by yellow marrow with age. Another primary organ is the thymus. This is where T cells maturate. And the thymus is located in the anterior superior mediastinum. So it's just behind the sternum and in front of the pericardium. And it extends from the lower parts of your neck, so above the manubrium, and down to the level of the fourth costal cartilage. And it's largest during childhood, and then gradually it shrinks with age. So important primary organs, bone marrow and thymus. Now we have secondary organs. We have lymph nodes, and these filter lymph and house immune cells. We have the spleen, which filters blood, removes pathogens, and recycles red blood cells. Now, when we talk about immune cells, we're essentially talking about white blood cells like lymphocytes and macrophages. Now, the lineage to arrive at these different cells, to understand it, you need to have this comprehensive idea of how blood cells or white blood cells evolve, where they come from. So it all starts with hematopoietic stem cell, right? That then becomes two different lineages. We have the myeloid progenitor cell and the lymphoid progenitor cell. Now, the myeloid then differentiates into megakaryocytes, which turn into platelets, eosinophils, basophils, erythrocytes, also monocytes that then differentiates into dendritic cells and macrophage. And we have neutrophils. But in the lymphoid progenitor cell lineage, we get the T cell, the B cell, and the natural killer cell. And the B cell also have these plasma cells. Now, in the course on hematology, this map here will become the main study, an object of main study. However, right now, just orientate yourself with it. Now, we have something called innate immunity, which means we're born with it. And here we divide it into our first line of defense and our second line of defense. Now, when we talk about the first line, we have physical barriers and we also have chemical barriers. In our physical barriers, we have skin and mucous membranes. For our chemical barriers, we have enzymes in our saliva and we have stomach acid. So, for pathogens to enter our body, we first have our skin, which repels it, mucous membranes, which catches it, enzymes that degrade it, and if it makes it all the way into the stomach, we have a very acidic environment in our stomach, destroying a lot of these microorganisms. We also have our second line of defense, but this is a non-specific immune response. And it's quite high yield to remember that it's non-specific when we're talking about our innate immunity. This is important when we move on to our adaptive immunity. Now, these non-specific immune responses include phagocytosis by macrophages and neutrophils, inflammatory response, natural killer cells attacking abnormal cells, and we have our complement system, which enhances pathogen destruction. And here we have the illustration of these three different types of cells. If you remember from the myeloid lineage, we have the neutrophil and macrophage. And lymphoid, we have the natural killer cell. Now, the neutrophil is a multi-lobed nucleus, or has a multi-lobe nucleus with granulated cytoplasm containing antimicrobial enzymes. The macrophage, which comes from the monocytes, has a large irregular shaped, or is a large irregular shaped cell with a oval or indented nucleus and has an abundant cytoplasm. The natural killer cell is a large granular lymphocyte with a round nucleus and cytoplasmic granules containing perforins and granzymes. Now, moving on from our innate immune system, we have our adaptive immune system. And characteristic for it is that it is specific and targets specific pathogens. If you remember, compared to our innate immune system, in our second line of defense, we had non-specific immune responses, like phagocytosis by macrophages and etc. We also have memory. So our adaptive immune system remembers past infections, and this aids in faster future responses. Key components here include our humoral immunity, so our B cells. These are mediated. So this humoral immunity is mediated by our B lymphocytes. And these B lymphocytes produce antibodies that circulate in the blood and in our lymph to neutralize pathogens. And we also have our cell-mediated immunity, characterized by our T lymphocytes, T cells. We have here cytotoxic T cells that destroy infected and abnormal cells. And we have helper T cells that coordinate immune responses. So just briefly, our B cells, right? They recognize antigens that sit on the target cell, and it undergoes activation. Usually there is a T helper cell involved in the activation of a B cell. And when that happens, this B cell can differentiate into plasma cells that secrete antibodies. And these antibodies, the role of these antibodies is to neutralize pathogens by binding to them. And then it also helps in activating the complement system, which leads to the destruction of these pathogens. We then have our T cell or cell-mediated immunity. And here we have, as mentioned, these cytotoxic T cells, also called CD8 plus T cells. And these directly kill virus-infected or cancerous or otherwise abnormal cells by inducing programmed cell death called apoptosis. Hence the name cytotoxic. Then we have these helper T cells, and they act as regulators of the immune response by releasing cytokines. And they also, as mentioned, are involved in activating our B cells and also activating our cytotoxic T cells and macrophages to enhance their different functions. Now let's just dive a little bit deeper into the B cells. So let's talk about antibodies and their functions. Now we have a detailed course on immunology where we talk about all of these different cells in great detail. But here is just an overview and some key anatomic structures. So, like we mentioned, these antibodies are produced by plasma cells. Plasma cells are activated B cells. The structure of it, it's a Y-shaped molecule with variable regions binding to specific antigens, as you can see illustrated here. And as we mentioned, some functions include neutralizing pathogens, something called opsonization, so marking pathogens for destruction, and of course, activating the complement system. But let's dive a little bit deeper into the structure of this antibody. Now, first of all, let's clarify, what are antibodies? Antibodies are also called immunoglobulins. So if you have come across IgA or IgG, some med students think that these are different things when talking about antibodies and talking about immunoglobulins. They are the same. We will talk about why these different types exist in our immunology course, but here you just need to know that there are different types of immunoglobulins. IgA, IgG, IgE, IgM, etc., etc. Now, these are specialized proteins produced indeed by our plasma cells, which are activated B cells, and they are produced in response to the presence of antigens, so foreign molecules or a pathogen like a virus, bacteria, whatever. And their role is they have a key part of the adaptive immune system because they target, like we said, specific pathogens to neutralize or mark them for destruction. So we keep using the word specific because we're talking about the adaptive immune system, which is specific. And therefore, these different types of immunoglobulins come about because they are specific for certain pathogens. Now, what is the structure of this antibody or of these antibodies? They have a very characteristic Y-shaped structure, as you can see illustrated here. And it has two main components that are high yield for you to know. We have a variable region and we have a constant region. So the variable region, we're talking about the tip of the antibodies. And the tips of these Y-structured antibodies are unique for each antibody. These regions bind specifically to a particular antigen's epitope. And an antigen's epitope is a small, recognizable part of that antigen. Then we have the constant region, and that's the stem of the Y. And it is less variable and determines the class of the antibody. So when we talk about the class of antibodies like IgA or IgG or IgD, for example, we're talking about something that is defined here at the stem of the antibody. And this region interacts with immune cells and other molecules to carry out different immune responses. And as you can see illustrated here, you see 4 structures. 1, you see 2, 3, and 4. And each antibody, okay, each antibody has 2 heavy chains and 2 light chains, and they are held together by disulfide bonds. So from the point of view of anatomy, remember, variable region, the tip, constant region, the stem. The stem determines the class of antibody. The tip is specifically structured to bind to a particular antigen's epitope and is unique for each antibody. Furthermore, each antibody has 2 heavy chains and 2 light chains, and they are held together by disulfide bonds. And hopefully you will never mix up antibodies and immunoglobulins, knowing that, oh, we're talking about the same thing. When we say antibody, we're talking about the entire structure here. When we say immunoglobulins, we know that we are referencing the stem of the antibody. That will determine which class it is. So it's both the same thing, but it's also a more specific part of that thing. And you don't need to get caught up on listing all of the different classes like IgA, G, IgM, IgE, etc. You will get plenty of that in the immunology course. But we can mention, for example, IgE is a class that is very much involved in allergic reactions and in our defense against parasitic infections, etc. IgG is the most abundant one that we have and provides long-term immunity, and is also the one that crosses the placenta to protect the fetus that is growing. So the differentiation of different types of immunoglobulins and classes has different indications and consequences and where they actually function and what they do. But hopefully now you got a brief overview. Now let's briefly talk about the lymphatic system. So when you study the early parts of medicine, you'll learn about the circulatory system, like veins and arteries. You'll learn that arteries is where blood is pushed towards the body to deliver oxygen, and veins is where blood is returning back to the heart. Let's keep it simple. But we also have the lymphatic system. And as you can see in this illustration here, it runs throughout the body. And it kind of looks like a part of our circulatory system. Now, the components of the lymphatic system is the lymphatic vessels. So this is where lymph is transported. And lymph here is fluid containing immune cells. Then we have lymph nodes. And this is where lymph is filtered and where pathogens get trapped. In these illustrations, you can see these more thicker parts, right? Here, you can see it here, 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 etc., etc. If you've ever felt your neck, you know that you have lymph nodes that enlarge when you have a infection, for example, near your throat area. And why is that happening? Because there is an infection. So a lot of white blood cells and antibodies are transported there, and there is an inflammation going on, so it swells up. So when we say lymph, we're talking about something that contains white blood cells and antibodies. So key distinctive things involved in our immune system. Now the function or functions of the immune system include draining excess fluid from tissues, also filtering pathogens, and supporting immune cell activation and transport. So draining excess fluids essentially means that if you have an abundance of volume in your circulatory system, where we talk about veins and arteries, if we have excess fluid, some of that gets drained back into our lymphatic system. But we talk about that in depth in our physiology course. So again then, the immune response. We have a primary immune response, which occurs during first exposure to a pathogen. And it's a slower response with lower antibody levels. Then we have secondary immune response, which occurs during subsequent exposures. It's faster and stronger because we now have immune cells or memory cells. Now, what's the clinical relevance here? What are some immune disorders? Well, we have autoimmune diseases. This is where our immune system, it's attacking our own body. And some diseases here include rheumatoid arthritis and lupus. We have allergies, so overreaction to harmful substances like pollen and food allergens. So for some reason, something enters our body that's actually harmless. It doesn't damage our body. It's not a bacteria or virus that wants to be invasive and destroy structures. This is where we need an immune response. When we talk about allergies, we're talking about an overreaction of our immune system to something that's actually harmless. But the key thing here is the immune response. If it's uncontrolled or exaggerated or just, unnecessary somehow, it still leads to clinical symptoms. Like you get an urge to itch, scratch yourself, or you have fluid running out of your nose, let's say, or your eyes when you're exposed to some pathogen or to some harmless substance like pollen. And that can be in of itself a problem. Because imagine you have this urge to scratch yourself and you start doing that, and then you destroy your skin, you start bleeding, and then that becomes the breeding ground for more inflammation. And then that just continues on and on and on. And you can think about, if you've yourself experienced allergies or if you know anyone with it, that it can take a life of itself, of its own. So that's one example of our immune system leading to different types of problems. Another one is indeed autoimmune diseases, and this can be very fatal. So this is for some reason our immune system has decided to target something that's actually a crucial part of our normal structure. For example, we have between joints in our body, we have something called synovial fluid. And essentially, it just helps this bone and this bone from hitting each other, which would be excruciating and painful. Now, what happens is that for some reason, our immune system has decided to attack structures in the synovial fluid, which over time leads to inflammation and destruction of this area. And this is, very briefly and simply put, the development of rheumatoid arthritis. Immunodeficiency, so a weakened immune system, like those found in HIV, for example. Because in HIV, the virus uses our T helper cells. If you remember, our T helper cells play a significant role in our immune system, helping in activating immune responses, etc., and signaling, etc. Now, in HIV, the virus slowly destroys these T cells. So they become less and less and less. And as that happens, our overall immune system goes down and we become immunodeficient. And this creates a breeding ground for very opportunistic pathogens that would normally be kept at bay. They now become ferocious and dangerous. And you get widespread infections, which will eventually even take the person's life. So now I kind of gave you an idea of how HIV continues to the development of AIDS. Transplant rejection is another clinically relevant part of the immune system. So this is where our immune system attacks transplanted tissues. So something is given to us, like a new organ, and our body recognizes this as a threat and starts rejecting it by attacking it. So you can see that our immune system, however wonderful it is normally, and its main function is to defend us against threats from the outside, it could make errors. It can overreact. It can be destroyed and weakened, etc. Now, vaccination and immunization. So what's the purpose of it? It's to stimulate our adaptive immunity to provide protection, like using live attenuated vaccines like in measles, inactive ones like in polio, mRNA vaccines like in COVID. We have herd immunity as a concept which protects vulnerable populations throughout widespread vaccination. Now, why it works in general and the theoretical idea is good is because what we do is when we have understood the immune system and we learn about plasma cells and we learn that, oh, our adaptive immune system has memory, what we then do is manipulate our body to strengthen its immune system by introducing a part of that pathogen. So if you go through life and a pathogen is presented to you, your immune system should react by finding it, destroying it, but at the same time, remembering by producing antibodies for the future. So the next time that pathogen comes, you have an abundance of these immune cells immediately, these antibodies immediately attacking it and destroying it. But what if the first exposure can be fatal? So the very first time you come across measles, let's say, or whatever, it could actually kill you or kill a lot of people in a population. So what we do is we say, okay, we'll take a little bit of part of that virus or that pathogen, enough to stimulate this person's immune response, but not enough to actually create a massive response leading to potential fatal outcomes. So we will just provide a little bit of it. And the idea then is to have our immune system respond to it and go through its automatized structure by producing these memory cells. So that when you go through life and that one time you're actually exposed to that pathogen for real, you already have a bunch of immune cells ready to attack it. So that's the concept of vaccination. And we differentiate between live and inactive and mRNA because, for example, in live attenuated vaccines, these vaccines use a weakened form of the germ that causes the disease. So you can, again, like we mentioned here, you still trigger a strong immune response, but you do not cause severe illness in people who are healthy. When we have an inactivated vaccine, like in polio, these vaccines contain the virus or whatever bacteria that have been killed or inactivated. So they cannot cause disease, all right? They can't cause the actual disease, but they can still stimulate an immune response. Now, in diagnostic tools, so we do blood tests, of course, to measure immune cell counts and antibody levels. We have allergy testing, where we identify triggers of hypersensitivity reactions. We can use imaging, so CT scans for lymph nodes and spleen evaluation. And also flow cytometry to analyze immune cell populations. Now, let's try to put this in some clinical context and try to tie the knot from the entire lecture. So we talked about our adaptive immune system. We mentioned B cells. We said that these plasma cells, which are activated B cells, produce these things called antibodies. And we mentioned that the antibody has two main regions. We have, if we cut it in two, we have the variable region, the tip of the antibody, and we had the stem of the antibody, which was the constant region. And we said that the constant region decides what kind of a class this immunoglobulin has. Is it class E, or is it class A, or D, etc., etc.? Now, you may think, why is it so relevant in knowing these details? And hopefully, in about a minute, you will know why. At least one example of why. So, the immunoglobulin E is primarily involved in allergy testing. Because what is the role of immunoglobulin E in allergies? Well, in allergic individuals, what happens is that the immune system is overreacting to harmless substances called allergens. Not pathogens, allergens. And the response is by producing excessive immunoglobulin E. Now, these immunoglobulin E antibodies, what they do is they bind to receptors on other cells called mast cells and basophils. So we can type out B here and M here. And these are immune cells involved in allergic responses. So upon subsequent exposure to the allergen cross-links immunoglobulin E molecules on these cells, causing these cells to release whatever they contain. And in this case, we're talking about the release of something called histamine and other inflammatory mediators. So, going a bit deeper, these mast cells, for example, we find them in tissues, especially in mucosal and connective tissues. And what they do is they release histamine during allergic reactions and during inflammation. The basophils are cells that are circulating in the blood, and they also release histamine but are less abundant than the mast cells. And so what happens when histamine has been released? Well, histamine plays a key role in causing vasodilation. So the dilation of your blood vessels, increased vascular permeability, and also smooth muscle contraction. All this during immune responses. Because why is this clinically important? Because histamine getting released from these mast cells upon activation from the immunoglobulin E, right, is that histamine normally is a key immune mediator that helps our body to respond to injuries, infections, or whatever. It has a lot of important physiological roles, like in wound healing or neurotransmitter function, or generally our inflammatory response system, etc. But why are we talking about it here as a problem, this excessive activation of immunoglobulin E, mast cell, histamine link? Why is this a problem here? Because we mentioned the effects of histamine, like vasodilation and increased permeability, like smooth muscle constriction. And why are these two problems? Because what do you get when you have vasodilation and increased permeability? You see symptoms of swelling, redness. What happens is that excess fluid leaks from our blood vessels into tissues. And then it leads to symptoms like nasal congestion, hives, or swelling, angioedema. What about the part where histamine leads to smooth muscle constriction? Well, in the lungs, for example, histamine causes something called bronchoconstriction. So the constriction of your airway, essentially, and that leads to wheezing, shortness of breath, etc. Now, hopefully you understand that we're talking about asthma. If you've touched upon emergency medicine or heard about it in a movie, maybe you've heard of anaphylactic shock or anaphylaxis. So imagine you're exposed to something completely harmless, pollen. That leads your IgE to overexpress, leading to mast cells releasing a bunch of histamine, leading to bronchoconstriction, tightening your airways, leading to the inability to breathe. And then that leads to, indeed, rigor mortis. So, in extreme cases, now we took quite an extreme case. Most people dealing with allergies is typically not this extreme. But in extreme cases, widespread histamine release can cause indeed a sudden drop in our blood pressure, airway closure, and shock. So it requires immediate treatment. So hopefully here you got an idea. We focused on histamine because it is essential for immune defense and normal physiological functions. But in allergies, an overactive or inappropriate release of histamine leads to the symptoms that can range from mild hay fever to severe anaphylaxis. And luckily we have treatments, something called antihistamines, and these block the histamine receptors to reduce these effects. So in summary then, the immune system defends the body against pathogens and maintains health. Some key structures mentioned include macrophages, neutrophils, natural killer cells. Then we went on to our adaptive immune system talking about B cells, plasma cells that are activated B cells. We mentioned our T cells. We had T helper cells, CD4, CD8, our cytotoxic T cells. We talked about the Y shape of the antibodies. We talked about the variable and constant regions of this antibody. And we also mentioned types of immunoglobulins and much more. So please do go back and revise. Innate and adaptive immunity work together for effective defense. Immune disorders can arise from overactivity or underactivity. And that's the end of this lecture. Continue now to chapter 13.